I'm using my Apple TV, my TV and my iPad for my presentation. How I normally do it in my classroom, I will have the students do a bell ringer, and after that, I will go over the objective and the agenda for the day. And this is normally where I'll pull up my presentation on Notability. I love Notability because it allows me to write on here and things like that. For the lesson today, we're learning about DNA structure and function. I will then go on to talk about our anchor piece for the next few lessons, which will be sickle cell anemia. I'll just explain to them how just the changing of one base pair in our DNA sequence ultimately changes how the protein structure is changed and the function ultimately changing how it affects our body. Then I'll open up the video.
Sickle cell anemia is a genetic disease that affects hemoglobin, the oxygen transport molecule in the blood. The disease gets its name from the shape of the red blood cells under low oxygen conditions. Some red blood cells become sickle-shaped, and these elongated cells get stuck in small blood vessels so that parts of the body don't get the oxygen they need. Sickle cell anemia is caused by a single letter change in the DNA. This in turn alters one of the amino acids in the hemoglobin protein. Valine sits in a position where glutamic acid should be. The valine makes the hemoglobin molecules stick together when oxygen tension is low, forming long fibers that distort the shape of the red blood cells, and this brings on an attack.
Okay. And then I'll go on to say how we're going to be learning residual dogma again, and how you saw that the changing of one base pair in the DNA strand ultimately changed the structure and function of the protein hemoglobin for red blood cells for the carrying of oxygen. And I will tell them in order for us to understand what went wrong, we need to understand and learn the structure of DNA or where mutations can happen. So I'll pull my Notability back up, my presentation, and I'll go into actually before I get to that, I'll probably ask anyone, do you know what DNA stands for? Now I'll get responses from students by raising their hands, and then I'll pull this up. They will have along some guided notes with them for the presentation. I usually like to give guided notes or a votable same thing like that. Sometimes have them write it all down, but just depends. And then I'll go on to say how functional DNA is needed to make proteins, which ultimately gives rise to our characteristics, which are our traits, such as hair color, eye color, and skin color, and things like that. I'll ask them, do you know where DNA is located? And a lot of times they will say, hopefully in the nucleus, and look at that, but And I also asked them, is DNA located in nucleus in prokaryotes or eukaryotes? And then that's why I'll have the recapping of prior knowledge of the location of DNA. And then hopefully they'll say in eukaryotes, and for prokaryotes it's located in cytoplasm. I'll go into say, what is the structure of DNA? Previously, we learned about biomolecules, so I will ask them what are the monomers of proteins. Ultimately, they'll say amino acids, and then I'll say what makes up an amino acid and things like that. They have previously learned about nucleotides and what makes up a nucleotide, but I'll recap over it, just a refresher for them. And in this case, I will re-show a nucleotide. I'll indicate to them what is the phosphate, the pentose sugar, and the nitrogen base, and how all of these nucleotides are going to come together to form a DNA structure, DNA strand. I will go on to say we'll learn about 3 scientists who found out the structure or indicated the structure of DNA strand, learned that it's a double helix. So I like to say it's like a ladder that is twisted. And so the sugars and the phosphates makes up the sides of the ladders, and the nitrogen bases makes up the middle or the steps of the ladder. And then I'll usually say these are our sugars and our phosphate groups. These are our nitrogen bases. And these are like our steps of the ladder. I'll also talk about complementary base pairing. A lot of them have previously learned about complementary base pairing, but just to recap, I'll let them know that for nitrogen bases, we always have 2, and A always pairs with T, G pairs with C or at, and then GC. I'll let them know there's 4 nitrogen bases, and just to kind of recap, or just to see, well, do they know anything about RNA? I'll ask them which base is not in RNA, and things like that. I will indicate to them that these bases, these nitrogen bases, are held together by hydrogen bonds. So 3 or 4 hydrogen molecules bonded together. And then I'll go into complementary base pairing. I'll remind them how when we have a strand of DNA, A is always paired to T, and G's are always paired to C's. So I have the students as a whole, as a big class, tell me what are the complementary base pairs to the strand. So they'll tell me, oops. So they'll say T, hold on, let me switch hands, sorry. Crap. So they'll say T, A, C, G, A, C, G, T. Okay, and I'll explain to them how there are always the same amount of A's, there are T's, same amount of G's, there are C's, because they are equal pairing. And so I will say if there are 35 A's, then there are how many T bases, and I'll say 35. And then I'll go into saying there are actually one person that actually truly discovered the structure of DNA. And I'll say her name was Rosalind Franklin. She actually discovered the structure of DNA using a special type of X-ray. And she determined that it was double helix. And the person she was working with, he actually took her information and gave it to Watson and Crick without her knowing. And then they used the information to create a model of the DNA strand, the DNA double helix. And this is Watson and Crick and their DNA model right here. And then I'll say how they actually got the Nobel Peace Prize, and ultimately she ended up passing away. And now she's starting to get the credit that she deserves, but ultimately she did discover the DNA model and not them. Okay, so after this time, I'll tell them, hey, take out your pieces. We're going to construct a DNA model. They previously already cut up the pieces like 2 days prior. And then I'll show them an actual model of the final product. And then I'll give them like a couple of minutes to construct. Well, I'll construct it on the board. these in big pieces, and then I have them construct it for a few minutes and then consult their partners, and then I'll display the answer key. Then I'll display the answer key on the board, most likely using this picture, and I'll zoom in. All the while I'm playing music on Pandora to keep the kids on task, and also helps them a little bit keeping focus. And also actually it's a noise monitor, because if I can't hear my music, then they're too loud. Sorry. And then after that, I have them clean up, and then it's time to go. So that's my presentation, or my technology used for my DNA presentation.